Lions Rock Productions.
This is Jay Moore.
This is Greg Cruz.
This is Dan.
This
Stone.
is Dexter from The Offspring.
Nathan
This
East.
is Sebastian Younger.
This is David
This
Lab.
is Stuart Copeland.
This is Mick
I'm
Gillette.
Krista
This is
Vernoff.
Dr. Bob Greenberg.
I'm Laird Hamilton.
This is Rob Bell.
Hey, this is John Leon Guerrero.
Hey, I'm Mark Valley.
Hey, and this is Pete A. Turner.
This is Attila Hajdu from Stelvio Therapeutics, and you're listening to The Break It Down Show.
And now, The Break It Down Show with John Leon Guerrero, Mark Valley, and Pete A. Turner.
I'm here on the road with my man Paul. Say hi, Paul.
Hey, folks.
And Paul is a long time friend of mine, a warrior, a combat guy, an Air Force master, a genius, but most importantly, he makes the best paella on the planet. And yes, he agrees. And so he's sitting next to me, and unfortunately, I'm sharing a microphone with him, so it'd be hard to hear you, so just getting close. But yeah, we're sitting across from Attila. Now, if you don't know Attila's work, you can go check out the episode of Popping the Bubbl that I've attached. For sure, check that out, because we talked to you back when you were with IndieBio at the accelerator there. And what they do is they accelerate biotech companies. And I could try to explain what you guys do, but this is why you're here. So could you just blow everybody's mind in about 2 or 3 minutes?
Sure. So thanks for the opportunity to have a conversation. Really appreciate that. So there's really no reason why people should be dying from cancer today, especially given the advances in medical technology and also given the fact that we've developed a technology that will usher in new treatments that can cure cancer. And so what we do is we discover and develop novel therapeutics and identify targets that are druggable through our innovative platform technology called microscopic imaging of the epigenetic landscape. And so basically it is a phenotypic screen using epigenetic markers. And what is epigenetics? Epigenetics is a control mechanism for gene expression. And so these marks exist in every eukaryotic cell. And what we do is look at the cancer cells and screen them using our platform technology and identify changes in the epigenetic landscape. And so what we're looking for it's an imaging technology.
Okay.
So what we're looking for is drugs that induce a cancer cell to differentiate from a cancer-like state into a non-tumor state. So this is the most fundamental secret of human developmental biology, which is how does a stem cell turn into something else? Like how does a stem cell turn into hair, or how does a stem cell turn into eye? So basically what our technology is doing is how does a stem cell, or what drugs cause this cancer stem cell to turn into a non-tumorigenic cell. So it's a cure.
Right.
And so we started off in glioblastoma because that's where our experience was. And we have an associate professor of neuroscience, who's our chief scientific officer. His name is Alexei Tursky. He's over at Sanford Burnham Medical Discovery Institute, and he's been working on the technology for a long time now. And we've got a world-class team of advisors, David Andrews at Sunnybrook Health Sciences Center in Toronto, and David Spanner, who's a medical oncologist. And we're surrounded by an infrastructure of the brightest, smartest people in the world. And so we've proven our technology through a proof of concept study where we screened a library. And I think this is where we last talked. We screened a library of small molecules and found a number of them that differentiate glioblastoma cancer stem cells into non-tumorigenic cells. And so in glioblastoma, the reason we chose that is because, first of all, it's a large unmet need. Most patients die from their disease in less than 12 months.
Like 98%, right?
Yeah.
Yeah.
And so you're probably familiar with some of the U.S. politicians that have been impacted by cancer. And the reality is that cancer impacts all of our lives. There's no one that isn't impacted by cancer. And so Joe Biden's son was impacted, obviously,
right,
by glioblastoma, and John McCain as well. And so, yeah, so the reality is he's going to die, and he gets chemotherapy, he gets radiation, he gets surgery. But at the end of the day, that's only going to give him maybe a month or two survival benefit.
Right.
But the reason we chose glioblastoma is nothing's been done in 40 years. So if nothing's been done in 40 years, that's what really inspires us, and the passion behind Stelvio is that I lost my mom from breast cancer. I worked for the largest drug company for 15 years, and I couldn't do anything for her.
Right.
And my dad died of cancer. He had prostate and pancreatic cancer. My wife, her mom died of ovarian cancer when she was 11 years old. So imagine her father raising the whole family.
Yeah.
And so I have a tremendous amount of respect for him. And he actually just died a few years ago from cancer. So somebody has to do something about this. So that's why I started Stelvio. And so it's a bit longer than I think 3 minutes, but you kind of get the problem and the solution and I think the passion behind it. I think that's 3 things that people really want to know about. And so what's exciting also is that we're raising a Series A financing of $5 million. We have $1 million committed from a lead investor.
Right.
And our company is now valued at $20 million. And so it's a big step up from where we were when we last talked.
Yeah.
When we were in the basement at IndieBio and trying to establish ourselves and sort of.
But it hasn't been that long either, though. I mean, it's been over a year, but not much more than a year, and not even 18 months, I don't think, since we talked. Mm-hmm. And it is an incredible advancement. You said that glioblastoma was the start, but have you moved outside of that particular discipline for cancer, or is that still like the route for you guys?
So that's basically we put together a very elaborate discovery and development plan. So our first tumor type that we're going after is glioblastoma, because that's where the experience is. Although our technology is agnostic to tumor type, meaning we can go after multiple tumor types. So we're sitting here in Johnson & Johnson Innovation, JLabs. So we're a JLabs resident company. And so everyone knows Johnson & Johnson. You know Johnson & Johnson. Sure. And so they have a pharmaceutical company that they own called Janssen. And so Janssen, interestingly enough, they have a very similar mission to what we have, and that is to deliver medicines of value to patients that need them. So the tumor types that they're interested in are things like lung cancer, prostate cancer, and hematological malignancies or blood cancers. And so we're actually in discussion with them, the Janssen R&D team and their discovery sciences people, and because we're part of JLabs in the same building as Janssen, we have obviously access to those people, and so we're putting together a plan for T-cell malignancies. Everybody has a CAR T-cell therapy, which is chimeric antigen receptor, which is basically taking the body's own immune system and using it to tackle the cancer. So the problem with a lot of cancers is that they're very invasive. The cells go into stealth mode, so the immune system can't attack those cells. So this therapy, you probably heard through the news that Novartis and Gilead have a couple of these therapies, and they're curative and in lymphomas like non-Hodgkin's lymphoma, but they're also expensive. Novartis therapy is $475,000, and Kite was just purchased by Gilead for $13 billion. So we know that Janssen are interested in that area, and I think they're behind everyone else. And so that's sort of the hot topic of where we're going with them. And also, it seems like there's opportunities to validate what they're doing, because with our imaging technology, we're able to see things that they can't see. Because we're down to the single cell level. We're segmenting the nucleus, extracting the features. We're down to the level of chromatin organization. If you think about chromatin, that's basically the unit of life. And everybody thinks, well, DNA it is. But chromatin actually organizes the DNA into chromosomes.
Right.
And then that's where the epigenetics comes in, because there's only two kinds of chromatins: open, where the DNA is easy to read, the genes express, and then there's closed, right, where the DNA is hard to read, the gene's not expressed. So those are the areas where there's a lot of interest is those epigenetic modulators. So the readers, writers, erasers, and things like that, because they control gene expression. And the other thing we're doing beyond cancer is that we know that in neurodegenerative diseases like Huntington's, that the epigenetic changes occur months, if not years, in advance of the gene expression changes. So we can actually see in advance predict the onset and potentially look for small molecules that delay the progression of Huntington's disease. So it's another area we're looking at as well. But of course, we have to be focused on what we're doing. Because you only can do one or two things well at one time. So that's where we're really focusing right now on glioblastoma. We're raising the $5 million for one indication. And then, of course, as soon as I close the Series A round, I'm going to start the Series B.
Sure.
Which is going to be like much bigger round. That's a $25-40 million round. And the thing is, you can't start these things overnight. I mean, you have to build relationships with these level of investors.
So we have you on here partly because you guys are helping to cure cancer, and that deserves a lot of our attention. And you're using AI and machine learning to get to the answer. So this is what's really interesting, and I think the audience will get, because I expose them to all kinds of things. I mean, they got Paul they're getting exposed to, and he makes paella and goes to war and does Intake the Spy, and we had rock and roll guys. So they'll get why you're here, because you're improving life, you're investing time and people's money into making something that has taken all of your in-laws and your parents, and it's everybody is touched by cancer. When I go out and I do my crazy work and I'm in villages talking to elders and everything, we're always told like, oh, these are Afghans, they're not a united people, they don't get along. Like, well, they all love their kids and they all hate cancer. So I've got some common ground to find, right? And this is a big thing, and that's why let's talk about what you're doing here with AI and how this is going to change our lives, because you're working on this. But like you said, there's peer companies. There's 20-something plaques on the wall of other companies that are in this early state. How do you compete with the people that have the venture capital when you have so many? Because this is just one area where there's great ideas. There's a zillion other companies trying to develop some kind of path towards reduced cancer or no diabetes or autoimmune things. How is the competition?
Well, it's a good question. I think that we get asked this all the time, like what differentiates you and what is your secret sauce. So the reality is that there's a different levels of risk. There's regulatory risk, there's market risk, there's scientific risk, there's technical risk, there's a number of things. So the way you overcome these levels, and by the way, if it wasn't risky, then everyone would be doing it.
Yeah, right. Yeah.
And it would be easy.
Right.
So the fact is, it's not easy. Curing cancer is not going to happen overnight. So you have to have the smartest people in the world to do those things. The people that are willing to sacrifice, the people that are living in like hostels like I did in San Francisco looking for money.
Right.
Not everybody's going to give up their big pharma job to do what I did and leave their family for 8 months.
Right.
So that takes sacrifice, and not everyone has that courage. Not everyone has the intellectual capacity to come up with an idea like, how do we induce cancer cells to differentiate into normal cells, and then use the AI. You brought in the AI, which is interesting because what we're doing is taking pictures of the nucleus, so the segmenting the nucleus. So just imagine the Earth is a nucleus.
Okay.
Right? And then we're taking pictures of it. So imagine a satellite from space taking pictures of the Earth.
Right.
So, and we can see the textures, so the bumpiness, the gradient. So we can see the mountains, the valleys, the water, the blades of grass, those things. And then we can see the changes, right? And then the changes give us an epigenetic signature or fingerprint. So the AI, just imagine the seasons and the Earth rotating and all sorts of things, and snow and rain. So there's no way you can tell the pattern changes with the human eye. It's just no way. So that's where the AI comes in, because a machine can recognize those changes in the patterns and then see which drugs cause those changes. There's going to be some drugs that cause nothing to happen, but there's going to be drugs that cause a stem cell to go down a pathway. And it's not a simple pathway, but the pathway of differentiating into a normal, like, non-cancer cell. So in glioblastoma, that's something like a oligodendrocyte, which basically just sits there in the brain and doesn't shoot out more cancer cells. So that's the thing with the biggest problem in cancer is that it's resistance. So resistance to chemotherapy, basically patients lose response to treatment, and then they're left with no viable alternatives, and then that's it.
You can't stop it, right?
So what differentiates us is no one has the ability to use imaging in the way we use it by labeling the histones. We're down to the molecular level, and then imaging it to see differences as part of phenotypic screening to identify novel drugs. And so not even J&J can do that.
Okay.
So if they can't do that, Pfizer can't do it, they can't do it, we can do it.
Yeah.
And so we have this ability to visualize, and now we're doing this with live imaging. And the live imaging is, so remember the satellite taking pictures of the Earth, that's a still shot.
Right.
So now we have the ability to do this live.
Yeah.
So we can light up the nucleus of a cell, and through live imaging, we can actually see cells dividing. And then there's a principle called symmetric and asymmetric division. So it's actually very simple, because if you think of a cancer stem cell, it divides symmetrically. So it goes, now we have two cancer stem cells. So it's basically cancer making more cancer. So what we're doing is we're looking for drugs that induce asymmetric division. So that means basically one cell dividing into a normal stem cell, which basically we need, and that can differentiate and die. And that's the problem with cancer, is that is it's uncontrolled growth of abnormal cells that don't die. So cancer stem cells divide symmetrically, normal stem cells divide asymmetrically. So they're producing a normal stem cell and a differentiated cell that can die, right? So the differentiated cell is what makes eyes and whatever.
Right.
So that's what we want to induce, is those types of divisions, asymmetric cell divisions. So that's what the drugs do. And so as I said, I think the simplest way to think about it is that with our imaging of the epigenetic landscape at the microscopic single cell level, no one could do that. And we've got the IP around it. So even if somebody did want to do it, they couldn't.
You said a lot of big fancy words there.
Sorry, but that's probably why
No.
we've had a lot of problems finding investors, because I think that going back to when I first started trying to raise capital, I mean, I was asked to give the Elmo presentation, like Elmo
Right.
from Sesame Street.
Yeah.
And then that didn't work because it doesn't sound very credible. So I basically went back to just following my heart and my instinct, which is telling it like it is. There's a lot of science here, and the reality is I can try to sort of dumb it down, although that hasn't worked. So I'm actually now finding that it does narrow the sort of the investors that will understand.
Yeah.
And that gets us another point, though, is the reality is that we've had experiences where investors say, well, we want to take a active position and blah, blah, blah. And we want to understand, and we want to do this. It's like, I'm thinking, well, there's no possible
catch up
way you could do that in
Right.
your lifetime. So the reality is you can't add value other than giving us money. So if they're not willing to do that, which a number of them aren't,
Yeah.
then of course that's bye-bye. And so, but we did as I said, we did manage to find investors that don't necessarily understand, but the reality is they trust the leadership.
Yeah.
Because it comes down to the management team. Can you trust them? And, I have to be honest, like nobody gets the positions I've been in at GSK and by not being trusted.
Right. Yeah.
And those jobs don't just go to anybody.
Sure. Right.
And just like the military, right? Not everybody gets those kind of jobs. I mean, only certain people get those jobs. And so you don't get to lieutenant or corporal or whatever, just unless you actually do something to get there. So so the reality is So what investors should look at is, okay, who's the management team? Can they be trusted? Have they done it before? Are they competent? And then another thing you should look at is, if you're an investor, is who are the current investors?
Yeah.
So we'll have, once we close our Series A, we'll have a group of investors that will go, be a nice list. And hopefully J&J will be on that list.
Yeah.
So that's, even if you don't know anything about what we do, nothing, zero.
Right.
You go, okay, so smart management team, they're like, know what they're doing.
Sure.
Look at their investors. They got J&J backing them. Their science is going to cure cancer, and they're passionate. So those are the things that people like, if I were an investor, that's what I would look for. I wouldn't say, well, I want to understand your science and I want to blah, blah, blah. I want to take a roll up my sleeves and take an active role in your company. And it's like, really?
Yeah. Let's talk about your ribosomes. Yeah. You're right. You have a very specific and unique company and what you do and everything. And a lot of this stuff goes like the things that Paul and I did. We went through training together, and they're like, you guys are going to go help the Army understand what's going on with the people.
Mm-hmm.
And that sounds great. Like, okay, great. How are you going to train us to do that? Like, we're going to have some people come and talk to you about things.
Mm-hmm.
Like, they didn't know.
Mm-hmm.
And they also didn't know, like, when you go to show up, a commander doesn't know he needs you. I've got to sell myself to him. And you don't get that. You don't get access to the rides without trust. So you've got to go out, and I've got to be able to go look at a commander and say, I know combat. I understand what your playbook is. I haven't looked at your mission statement, but it's got these 4 elements in it, which it does. And if people don't believe that the cops that you're training are proficient, then you're losing. If, these things, so if you give me 3 weeks, you'll see what I do. And in 3 months, you'll look at me and go, Pete, you're the most valuable person I have. Because I know commanders love audacity. And so you're audacious. And then and 3 months later, when I see him, I'm like, you don't have to say it. I already know. We already know. We've already had the conversation on the value that I bring to you. But that only happens because I was able to build trust and then deliver.
Of course. Well, that's what the definition of trust is, right?
Yeah,
is delivering consistently. It's like one of the things I learned about talking to some people in the Army is that the commander, the person that is in charge of the troops, has to be the guy that would crawl through the ditch first.
Sure, yeah.
And that's the thing that all leaders, good leaders, should do. It's like they have to be willing to go.
Yeah.
If no one else would go, and so which I found pretty interesting. But anyways,
well, that thing, and the other thing that you do is something that we learned from Hilliard. It's a friend of ours. He's a screenwriter, and when they go out and pitch, they pitch movies and ideas to HBO or Amazon or whoever. And he's like, you don't come in and say, I'm curing cancer. You go, hi, my name's Attila, and both of my parents died from cancer. My wife, both her parents died from cancer. And you tell this story, so they're like, I need to know what you're going to say next. And you sell yourself, and so they're like, wow, what are you here to talk about? Like, well, I want to tell you about this story that I've got to do, or I'm going to take. So that might help, but it definitely, in terms of what they're doing, because it's very similar where HBO or CBO, whoever it is, there's 100 people like you coming every day, always looking, and how do you build that trust? And it's you open with yourself. You say, this is how you do it.
Yeah. Well, biggest question, you've obviously, you got the passion and you've developed yourself from nothing to a $20 million company. That's impressive in itself. Well, I'm just curious, if we were investors, which we clearly are not. Okay. But if we were, do you have something you can hang your hat on? What would you show them? Let's say they didn't have a great deal of background in medical research and whatnot. What would you show them? This is why this is so promising, what we are doing. Do you have an event like that or an example?
Sure. We actually have something that's never been seen on the planet by many people. And what that is a video. We have a video now where we're showing this video to investors. And the video is actually showing the live imaging of the epigenetic landscape. So we're actually showing investors that we can track, imagine 2 cells dividing, and going back to the symmetric division, we can see which ones are dividing symmetrically and which ones are dividing asymmetrically. And then we can follow the epigenetic changes in the landscape. So that's basically one of our key, you can actually see something that no one saw before. It's like, just imagine a video of a man landing on the moon.
Right.
I mean, it's the same thing, right? It's like, holy shit, like no one's done this before, and there's live evidence right there. And I've got it on my phone, and now it's we're developing different colors, so it's pretty interesting. Even if I just show one video and say this is what we do, because it's never been done before.
Do that raw audio.
Yeah. But in terms of audio, I mean, it is pretty fascinating to see that. And as I said, it's a major secret of human developmental biology, which if we unlock that secret in terms of the mechanism by which cancer stem cells differentiate into non-tumor cells, then that's a huge game changer in terms of improving the future for our kids. And I think that's really the simplest mission statement for Stelvio is we're just all about improving the future for our kids.
Let's talk about the future a little bit, because you have done so much in the year or so that we've known you. And I don't want to put you on the spot, and say you said on this date, but
this episode of the Break It Down Show is brought to you by Lions Rock Productions.
That's us.
We publish, evaluate, and develop podcasts just like this one, consult others to build their own, and create associated content and content marketing strategies.
So if you're launching or expanding your social media presence, your business or your personal brand, or if you just want to take your media presence to the next level,
reach out to us on Twitter
at Pete A. Turner
or at JohnLG69
at the Break It Down Show.
There's 1,000 ways to get a hold of us.
Now enjoy the show.
when we look at a cure from cancer, is it 10 years away? Is it 20? Can you even say yet? What do you think?
We don't know, and I don't mind being put on the spot, but basically I like the hardest questions. I mean, that those are the most interesting ones. And so basically what we can see now is if imagine these glioblastoma cancer stem cells in a dish.
Yeah.
And we put these drugs, which we've discovered and we've identified, we can see them differentiating into non-tumorigenic cells in 11 days.
Wow. Okay.
So the reality then is I have people calling me that know I'm working in glioblastoma, and they say, well, my daughter's teacher has glioblastoma. What can we do?
Yeah.
I know you're working in it, and the only thing I can think of is, okay, can we put them in the clinical trial?
Right.
Or do we go to find some neurologists, some oncologists, and say, listen, this is what we're seeing. We should try a small clinical trial to use these drugs. Because, by the way, the drugs we found are approved medications.
Already. Yeah.
And however, that's not our intent. Our intent is not to commercialize drugs that are already approved, which is called repurposing. Our intent is to discover novel, new therapeutics. And that's not to say, though, that companies are not interested in repurposing. If you talk to companies like Genentech, which is big in oncology, they're simply flat out say we're not interested in repurposing. And the reality is because they can't make money off of it. Because then it's like, well, how do you market aspirin, for example? Okay, aspirin. What do you take aspirin for?
Oh, my heart now.
There you go.
Yeah.
There's one. But what happens if I said to you, well, you can take it for mood disorders?
Oh, really? Oh, I'll take some of that.
Or whatever.
Fish oil pill this morning.
So there you go. So it's a bit of a nightmare for pharma. And aspirin is kind of a very general example, but you get what I mean.
Yeah.
Right? You can't make money on it. And so they say, and I think we talked about this the last time, is that is a problem, because if let's just say it costs $1 billion to develop a drug to get into a person over 20 years.
Sure.
The failure rate's 90%. So you're throwing away $900 million dollars, and these things just sit on a shelf somewhere. And if you ask those companies, well, can you let us do something with those? They'll say no.
Right.
Some will say yes, and some will say, well, and there are companies that have been successful in raising money on just a repurposing strategy. And so that's not out of scope for us, but it's certainly not the focus. A lot of this comes down to the medicinal chemistry later on that happens. So when you first discover a molecule, you have to increase the selectivity for its target. So think about a dartboard. You want to be able to hit that the center each time.
Right.
You don't want to hit the periphery because that's going to cause adverse events.
Right.
So you want it to be very selective. So when you first come up with a hit, it's not selective. So the medicinal chemistry increases the selectivity for the target, and also it increases the potency. So you want something potent. So that's very selective. So you can use a very low dose.
Right.
Because the lower the dose, the better. And obviously in terms of side effects, because all drugs, as you know, have side effects, which is another advantage of ours. Going back to the competitive differentiation, is that the doses that we're looking at are like probably 10 to the 3 level below therapeutic doses, because you don't really need
the full shot.
Because we're not trying to kill that cell.
Right.
So that's the big differentiator. Is that,
because there's so many things where we could, and we're running low on time, so we can't get into all of it. But one of the things that fascinated me before, there's a couple things, and I like to say this because it's like a monkey's learn something cool, and I do it. So I like to talk about how like your AI works like in 57 dimensions, but then breaks it down to 2 so we can understand it.
500 actually.
Now it's 500?
It's 5. I don't know what 57. Heinz 57. You have
my
food on your mind.
brain does not work at the level.
500 dimensions, because there's 250 dimensions per channel and 2 channels. So it's 500 dimensions, and then it breaks it down to 2, kind of like the break it down show.
Yes, like the break it down show. So there's that aspect, but there's also like the ability to tailor this solution to a specific person. It's not that everybody has finger cancer comes over here and take these radiation shots and take this poison pill. That's what you got to do. That's what you got to do. But you're saying you don't have to do that as much. You have this specific kind of cancer, and we're going to turn it off.
Basically, yeah.
That's incredible.
Yeah.
Turning off cancer, Paul. What do you think about that?
It's very much self-interest. Like you said, it's not touched by it, My mother, my grandfather. I just found out this morning, matter of fact, before I even met you, that a lifelong friend of mine is going to be dead within 2 months. So I wish you all the best of luck. It's our own self-interest.
Yeah, it impacts families too. I mean, the biggest thing is I have people talking to me saying, well, I've got a colleague or her husband got diagnosed with glioblastoma, and the family wants this man who's got glioblastoma has gone through 4 rounds of chemo to fly by himself to Florida through like a 4-leg trip. I'm like, pardon? And so you see, like, families get involved and it actually tears families apart when you have someone in the family with cancer, and it actually puts a lot of stress. It did in my family too. It my sisters and I were fighting, and my mom, we actually she lived in my home. I took her in.
Yeah.
I drove her to the chemo clinics, and they hooked her up to the chemotherapy, and I sat there with her and brought her home. And even just getting to a chemotherapy clinic, just imagine if you're living in Lethbridge, Alberta,
right,
and you have to go to a chemo clinic. What do you do? It's very stressful. And my father-in-law basically just said, I don't want any chemo.
Yeah.
Don't want it.
I turn my cancer off by turning me off. Let's just get to it. Yeah. The other thing about all of these things is no matter what the level of care and the passion is, at some point you're going to a system, and the system isn't rewarded on an outcome. Like, they don't get paid. They get paid regardless. And so it's not to put a negative thing, but you are fighting for your mom's life, and they're there doing their job. And that creates a lot of negative perceptions. And it's really, I mean, think about all the damage that comes from all of these things. My friend's dad just died a couple of weeks ago from cancer. And he's like, it's the hardest thing I've ever done. Like, you're raising kids. It's
that's pretty tough.
Yeah, pretty tough. So if this is the hardest, and I believe him, it's the hardest thing he's ever done. His dad passing, of course, is tragic, but these things don't have to be that way anymore. If we can get this thing figured out here, with the epigenetic markers and the painting and all these, I'm gonna stop saying fancy words because
that's good. That's good. You're getting it.
I like to do this. I like to say, for all of humanity, I'm going to speak for everybody. We want to thank you for doing this work, for making the sacrifice, for living in the hostels, for trying to build trust with people that are getting pitched from all kinds of angles. Thank you for putting up with that. Thank you for being away from your kids and not living in your home, Canada, and being down here in San Diego, although that's nice. Still not home. So thank you for doing those things. And you've always got a platform here you can talk from. I don't mean here at JLabs, of course here, but also on the Break It Down show. We want to have you back.
Yeah,
and
thank you so much.
Yeah, it's really fantastic. Anything else you want to say before we close it out?
Well, stay tuned. The next 6 to 12 months, we're looking to identify novel targets and therapeutics. And I think within 2 years, we'll have a lead candidate that we can partner with a pharma company that we can take into clinical trials. So that will be, and the exciting things about these types of treatments or cures is that oftentimes you don't have to go all the way to phase 3 clinical trials. Like I've personally worked with drugs that have been approved on phase 2 trials because they're just so
needed and
remarkable.
Right.
The effect is remarkable. They don't need like a large phase 3 randomized global prospective clinical trial. Like the phase 2 data is enough.
Yeah.
So people are hopefully going to hear more about us, and I'm sure, stay tuned, and hopefully we can do it again another 6 to 12 months, and we can talk about all the exciting developments.
Absolutely. So how do folks, there's a lot of people that are going to hear this and go, I understand half of it. And also, who are these guys? Tell them how to find you.
So we have a website. It's Stelvio-oncology.com. And that's one way to find us. Obviously, if you go to the Johnson & Johnson Innovation webpage, they do a lot of marketing for us. So if you go to Johnson & Johnson Innovation JLabs, San Diego, you can find us.
Okay.
And you can see pictures of the team and different people. And then obviously we do send out the odd tweet every once in a while. We have a Facebook page, which we really don't use. But best way to find us is here in San Diego with JLabs. And that's basically where we operate out of.
I love it. All righty. Paul, anything in closing?
No, thanks for having me.
All right. Thank you again, Attila, for coming on. It's great. Yeah, you can find him at stelvio-oncology.com. I'll put that in the notes. You're Paul from Paul's Paella. Yes, everybody should go follow them on Instagram and Twitter. I'm Pete A. Turner. John is John LG69. Mark Valley is Mark Valley. Paul's Paellas with an S.
Cool.
And thank you all, and we'll talk to you soon.